Good morning, everyone, and thanks for coming to the talk. I work in Katarucha Lab, whose main focus is at looking at pathways that could be exploited in controlling mosquito reproduction as a way to reduce mosquito population and further reduce malaria transmission. I am specifically interested in looking at lipolytic machinery in the mosquito that might be potentially important in controlling reproduction and infection. As you all are aware, Anopheles mosquitoes, the female mosquitoes, require a blood meal to initiate egg production. This is a process that takes about 48 to 72 hours to be completed. If the mosquito is mated, she'll be able to oviposit her eggs, and then she can go on to have subsequent blood meals. Usually, you could have about 3 to 4 gonotrophic cycle in the lifespan of a mosquito. But at the same time, blood meal ingestion also is a gateway for the transmission of malaria parasite for them to move on to the next stage in the mosquito. So once the mosquito ingests an infected individual, you would have malaria gametes, plasmodium gametes that will undergo fertilization in the midgut of the mosquito to form okinates, and these are invasive forms that will invade the midgut epithelium to establish oocyst. And this would normally takes about 7 to 14 days to develop into sporozoites, which would then migrate through hemolymph to get to the salivary glands, and they are ready for transmission to the next mammalian host during the next blood meal. So what we see is that the process of egg production and the establishment of plasmodium infection in the mosquito, these two processes are temporally and physiologically coupled. And one could think that processes or factors that could be important for egg development could also have an implication in malaria infection. And so one of such, what we find is that in blood, usually there are a lot of nutrients. There have been a lot of studies looking at the role of proteins in terms of egg production. And the focus of my talk and the project that I'm developing is looking at the role of lipids in egg development and then in the establishment of infection during these critical stages of the parasite life cycle. So while lipids are a major component of biological membranes, and among other functions, they are also mediators of intracellular signaling. So it is obvious to then ask the question, what is a possible role that lipids might have in these two physiologically coupled processes? Just looking through the lipid biosynthetic machinery in the mosquito, there is a conservation in terms of the pathways that are involved in lipogenesis, like the making of lipids, where you could have acetyl-CoA, which is a precursor molecule that could be obtained from amino acid or sugars, that will undergo further processes to form fatty acids, which could be coupled, to a glycerol backbone to form major lipids like diacylglycerol, and you could have further acylation of that to form triglyceride. What also happens is that you could have also lipids in blood, like fatty acids, that could also be transported via the membrane and conjugated to the diacylglycerol backbone to further this process. Because once these lipids are made, they are then loaded onto lipofurine, which is a major lipid carrier in insects. This lipofurine can then be transported to ovaries or transported to fat body, where they're stored and could be mobilized during subsequent other needs in the mosquito. So we are interested in looking at lipids, and so in order to address to dissect this, we took an unbiased approach where we fed adult mosquitoes blood-fed adult mosquitoes and then collected mosquito tissues like ovaries, fat body, and midgut. At the same time, we also have control mosquitoes. We then did total lipid extraction and mass spec analysis with collaborators at the Broad Institute. To summarize what we have from the mass spec data experiments, we observed that, like among the tissues in ovaries, we have over 40%, 400% increase in total lipid content. That is followed by fat body, where we see an increase in lipid content at 24 and at 48 hours post blood meal. We don't see much change in the midgut, but there is a significant increase in what we see at 48 hours. So for the rest of my talk, I'm going to talk about just two major lipids, diacylglycerol and triglycerides, which are very similar in terms of hydrophobicity. And so looking into this mass spec data, what we see is that when we look at midgut, in terms of percentage of total lipids, we have diacylglycerol is increased in blood-fed mosquitoes at 24 and 48 to a lesser extent, but then we have a significant increase in triglycerides. Whereas in fat body, we were even surprised by this data. What we see is that diacylglycerol is reduced in both time points, and similarly for triglycerides. But we have a tendency of an increase in these lipids from one time point to the next. So if we had done, say, 72, perhaps this might recover later. But what is very striking is that looking at ovaries, we see a significant increase in the levels of diacylglycerol, but then we see a decrease in triglycerides in ovaries. So this sort of suggested to us that perhaps this there could be a lipolytic machinery that is either breaking the triglyceride to produce diacylglycerol. And so we looked into the Anopheles gambiae genome to see if there exist any triglyceride lipases that could be mediating this process. And what we found is that there are two lipases, which I name here triglyceride lipase 1 and 2. Normally how lipases work, they would catalyze the removal of the first fatty acid, for example, in this case, to produce diacylglycerol, which can undergo further lipolysis to produce monoacylglycerol until you have the glycerol backbone released, and that can be used for subsequent processes. So the next question is, do they play a role in this process, in blood feeding? So the approach we took was to design double-stranded RNAs against TL1 and TL2, and these were injected into adult mosquito, one-day-old mosquitoes, and at 3 days post-injection, the mosquitoes were blood-fed, and we will analyze then the number of eggs that the female will produce 48 to 72 hours post-blood meal. What we found is that inhibition of TL1 and TL2 significantly reduces the number of eggs that the female will produce. So what you find here, each dot represents the number of eggs in each female. And when we do combine TL1 and TL2 injection, those females with incident effect is further accentuated. But it's very striking is that TL2 seems to be having a more stronger effect than the TL1. So then we decided to assess whether, in addition to observing a reduction in number of eggs, are these eggs viable? Are they good in any way? And so for that, we did fertility experiments in the same scheme. So 1 day post-injection of the double-stranded RNAs, we insert the females in a cage of males for 2 days, and then on day 4, they were blood-fed, and 3 days later or 2 days later, we provide them oviposition cups for them to oviposit their eggs, and within 3 to 4 days, we then look at viable offsprings, how many viable offsprings that emerge from these eggs. What we found is that in addition to, unlike TL1 where we don't see any effect on fertility, we see that TL2 inhibition significantly impairs fertility of these eggs. Very striking is that if you look at this TL2 population, or the mosquitoes, over 95% of them, each female would have about 100% infertile eggs, which is very striking. But we are somehow puzzled by the fact that in the TL1, TL2 injected mosquitoes, where we see a further decrease in number of eggs, we don't see this phenotype as what we see for TL1, for TL2 only. So we're still trying to understand what could be happening. These lipases have not been described before, so we're still trying to get our head around it. So altogether, these data suggest that diacylglycerol, which is the breakdown product of this process, or the free fatty acid that emerged from there, it's important for egg development. As I mentioned earlier, that egg production and Plasmodium infection are physiologically coupled. So the next thing is then to assess whether TL2, so we decided to move on with TL2 because TL2 is the one that has the strongest effect on egg production. And so we then tested whether TL2 inhibition would have an effect on infection. Same experimental scheme, but then instead of providing non-infectious blood, we provide an NF54 p-falciparum line that was generously provided by Carolina's Barrel's lab. And at day 7 post-feeding, we dissected these mosquitoes and counted the number of oocysts in each female. So what you see is that inhibition of TL2 seem to favor infection. So that would suggest that triglycerides are important for infection, which is opposite to what we see in terms of effect. What we see on egg development here, we've seen favoring infection. And if you probably attended Flaminya's talk yesterday, where she showed that from some laboratory and field experiments, we have observed that usually the number of oocysts that a female will harbor usually correlate positively with the number of eggs that she will produce. But that doesn't seem to hold in this situation. We are seeing instead an inverse correlation. So this is important in terms of when we think of transmission dynamics, because if we are trying to reduce mosquito population, we are looking for tools that will reduce mosquito population, but then we, at the same time, also favoring parasite infection that might have dire consequences on transmission dynamics. And so we have to be a bit cautious in terms of which pathways we will take further to explore in terms of controlling transmission. So to summarize, using an unbiased approach, we have identified that a lipid lipolytic machinery is crucial for reproduction and Plasmodium infection. We are now currently doing lipidomics analysis of these mutants to really ascertain that the tax levels are affected, because we would expect that in this mutant, the tax levels should be high compared to controls. The next set of experiments are also, we're trying to then rescue, if we can rescue this phenotype, like the fertility, by injecting either diacylglycerol or phosphatidylcholine, which is one of the main phospholipid in membranes, and DAGs are very important for PC synthesis. Again, the crucial part is then what is the mechanism by which this is happening, and how is this regulated. So with that, I would like to thank my colleagues and the entire lab and specifically Jamaica and Christine for their help in some of the experiments, our collaborators at the Broad Institute, and our funders. Thank you so much for your attention.
Questions?
Great talk with you. I was wondering if the impact on fertility that you see when you do the double treatment with TL1 and TL2 dsRNA is potentially due to a dose effect, or whether it's mechanistically interacting. Have you tried injecting the dsRNA as TL2 with something like dsRNA as GFP?
No. So we're thinking that there might be some dilution effect. That's something that we're thinking of. But thanks for your suggestion, like doing the TL2 and with the GFP. That could help us in a way.